being willing and ready and raring to go. Okay. So AP Bio, and this is our second day talking about enzymes. We did a lot of pair decking last class. We don't have so much to do today for that. I'm gonna try to make a really brief presentation on enzymes, enough so that you get the information that you need, especially to do this lactase enzyme lab that we're going to start today. So today you are going to get a chance to do some lab work on your own and to play with the simulation. It's kind of fun to play with. So first of all, before I forget, next Friday, no matter what else happens with the lab or anything else, next Friday is your unit test for enzymes, just for enzymes and the free energy graphs and how enzymes work and the active sites and rates and all of that. Okay, that's next Friday is your unit test on that, no matter what else happens. I don't want you having to do it after Thanksgiving. That's gonna be bad. And in the meantime, I'm going to finish up on enzymes today and show you a couple things and then get you started on the first day of lab collection. Okay, so you're going to do some collection of data today that's actually going to be towards your individual grade, the individual parts of your grade for this lab, which I will show you in a minute. Okay, so first of all, let's just talk about enzymes. Let's go back to that pair deck just for a minute. We don't have much left. I just wanted to show you a couple of things on there before we go on. So, find it. Excuse me a minute. How's everybody doing today? Kind of a rainy day. Kind of a strange day. Actually partial to rain. Maybe that makes me weird, but I like it when it's raining outside and I'm cozy and warm inside. I don't know about you guys, but that's how I feel about it. All right, so let me go to pair deck. All right. There we go. So again, we're gonna go straight to the most important slides on enzymes here. We're not gonna linger too long on them, but so if you need to do any lengthy note writing, you may need to do that later. Today, I'd just like you to participate in the pair deck so that I know you're on top of the things that we're talking about. What we'll do today, the stuff that you'll see today is going to remind you a lot of freshman year. This is going to be reviewed. That first stuff on energy graphs, that was new to you. All of that was new. That's why we needed to spend more time on that. But this, how enzymes work, I'm thinking you probably already have a lot of this under your belt already. So you're not going to find this as difficult. So we're not going to spend a huge amount of time spending lots of time on this, because I think it's going to be a lot more fun for you to go in and play with the simulation and get some data. Okay, I'm just going to go now. If you still need to join, you still can. Make sure that I have I took your attendance using this this last time. So as long as you were in it and participated last time, you're fine. But you should be participating just because it's less boring than just sitting there, right? Okay, so join us. All right, so I know we don't need to do any of this front stuff. You got all this. We did all this. We did all this. I know what I'm looking for here. We did all of this. Okay, that right there. All right. Okay, this picture, I like starting with this picture in terms of talking about an enzyme. The enzyme is the big blue thing, okay? And an enzyme is a protein that is specialized to speed up a chemical reaction. So this is the enzyme before it meets up with its substrate, which is this little red thing here. And it has a pocket. It has an active site on it that has all of the right characteristics in terms of shape and chemistry and ionic charge and pH to successfully receive that substrate, that particular substrate. Enzymes are very picky. And then once the substrate is in the active site, you see how the enzyme changes shape slightly, and that's called induced fit. It kind of gives the substrate a little hug. And then the substrate's chemistry is changed. In some cases, it's breaking it down into two parts, like the lactase enzyme that we're going to study today. That's an example of an enzyme, the lactase enzyme, that would turn this substrate into 2 bits, right? Break it down. There are also enzymes that take 2 bits and bond them together into one bigger, okay? So either way, that's how an enzyme works. How to do that? It does it by the active site orienting substrates together correctly so that they're more likely to form bonds, or to strain the molecule so that you're more likely to break bonds. There's all kinds of ways the active site does that. How many can you? I'm not going to worry about that. We're not going to worry about that. You know this picture, right? This is the same as the other picture I showed you. It's just in cartoons. This is an example of an enzyme that is taking 2 smaller molecules and changing their chemistry and then releasing them as a process. And the point of the picture is that the enzyme starts with a particular shape, and then once it's done, it reverts back to that shape. Enzymes are not consumed the reaction. Okay, this is important because this connects with the lab you're going to do. What are some environmental factors that you know might affect how well an enzyme works, or whether it's the right shape, or whether it's going to work best? What are some things that you can think of? Again, straight out of freshman year, you talked about this in your bio classes, I promise. It's just a while ago. What can you come up with on that? Yes, that's it. You guys are awesome. Temperature and pH, that's basically it. Other things too. There are some other molecules that, salinity is another one. Good for you for thinking of that one. That actually does matter, yes. Concentration of the substrate matters. I can see what you were trying to write. That's good, you got it. Yes. Heat can denature an enzyme. You're ahead of me. Excellent. If you have too much heat, that can denature enzymes. So that is one way in which temperature can affect an enzyme. Yes, you guys have got it. That's good. Good. Going? Right? So you got all those. Okay, so this is an important screen, and this will help your enzyme lab that you're going to be doing the next couple days. Each enzyme has an optimal temperature in which it can function. Optimal meaning best, right? So some enzymes, there are some enzymes that exist in bacteria that live in the boiling hot springs in Yosemite National Park. You've seen those blurping, bubbling hot springs out there. There are bacteria that can live in there. And that's because their enzymes are particularly adapted to be able to work at really high temperatures. Most enzymes can't. Most enzymes would denature at those temperatures, but each different enzyme has a different optimal temperature. This is the same for pH. There are some enzymes which work perfectly well in the acidic stomachs of animals, and there are other enzymes that need to be in more basic environments in other parts of animals. So each enzyme, depending on where it evolved and adapted, is going to work at different optimal temperatures and pHs. Okay, so now let's look at the salivary amylase enzyme, which is in the human mouth. It works in our mouths. Keep in mind, our body temperatures are about 36 degrees Celsius, right? 98.6 is normal Fahrenheit, but 36 degrees Celsius would be our body temperatures. So what do you think the curve would look like here for the enzyme activity of salivary amylase? Your chance to do some artwork. Right? Chance to do some artwork here. Go for it. Okay. Yeah, what you're really looking for here, what you're going to see in real life, are bell curves. You're going to see bell curves, yes. So the peak of the bell curve, if you really understand what's going on here, the peak is going to be at 36. So definitely not at 100 because that would be boiling hot and the inside of your mouth is not boiling hot. Right? Water boils at 100 degrees Celsius. So the peak would be, yeah, it would be more like that. That's it. Right about at 36 is the optimal temperature for salivary amylase because it works in the mouth. So the peak would be here, and then it would denature, if you think about it, at these higher temperatures, and it wouldn't work very well in extreme cold either, right? So yeah, that's a good one. Yeah, you got bell curves for this, but that's just to give you an idea of how that works. All right, let's keep going. Now let's try pH, right? Now think about pepsin, which is an enzyme in the human stomach. So I decided to get you to guess, what do you think the pH of the human stomach would be, and where would pepsin work best in terms of the curve of this graph? Where would it work? Where would it not work? See what you can do with that. Some more artwork here. Okay, let's see how you did. Yeah, that's good. So this peak is way back here in the acidic part. Yes, that's it. Yes, so you're going to get peaks that are not, this is basic up here, and your stomach is very acidic, so it would be more down there. Yeah, see, that's good. Trial and error is good science. There, that's very good. Yes, I like the different colors, right? It probably wouldn't work well here at 1 because in extreme acidity, most enzymes get pulled apart. Yeah, that's fine. That's a trial and error again. Once again, it would peak here because your stomach is actually very acidic. It's a rare place inside our bodies where the pH is not more neutral. The pH of your blood, of your bloodstream and other places, is right around here. Right? So those enzymes would peak here. Your stomach enzymes would peak back there, right? So good, you're getting the idea. That's fine. I think that might be, there we go. See? So here's some actual examples of some sine curves. So this would be a typical human enzyme. Most of those, like in your mouth or in your bloodstream, are working at a temperature of, typical human temperature of 37 degrees. But in terms of temperature, a heat-tolerant thermophilic bacteria, like the ones that would be in the Yosemite, boiling hot springs, they can actually have an optimal temperature that's much higher. And it has to do with their structure, and it has to do with their resistance to denaturing. There's a pH graph. So there's your stomach. See? It doesn't even work past a pH of 3 at all. Look at that. So that's kind of interesting. And there's your pH of your stomach. I actually had no idea it was that acidic. I thought it was more like 4, but yeah, your stomach is a very acidic place. And for trypsin, this is interesting. This is a good data point here. You might need to know this. This is an intestinal. So in your intestines, the pH is a little bit more than 7. It's slightly basic in your intestines. So that's a place where you have lots of enzymes breaking down fats and carbohydrates and sugars and proteins that are going through your intestine. That's an important data point. And I think that's it. Okay, that's all we need to do with that. All right. So now let's go to talk about. See, I'm going to share a video with you. That's an extremely nice video that explains how enzymes work in general. And it does it at the molecular level way better than the paradeck did. So I want to show you this. I also uploaded it for you if you want to go watch it later. So I'm just going to turn my sound off. I just want you to watch and listen. Okay?
Every single second, inside every living cell, thousands of chemical reactions are taking place. These reactions are performed by enzymes. An enzyme is a protein that catalyzes a chemical reaction. It initiates the reaction, speeds up the reaction's progress, and makes sure the outcome is always the same. These enzymes often work together to form longer pathways, such as the citric acid cycle, which which is a series of chemical reactions used by cells to generate energy from carbohydrates. The essential tasks of life, such as metabolism, protein synthesis, and cell renewal and growth, are all regulated by enzymes. The life-sustaining power of enzymes lies in the fact that they catalyze reactions in mild conditions of pH, temperature, and atmospheric pressure. The rates of catalyzed
Okay, I just wanted to stop that for a minute just so that you can look at the screen for a second so you know what you're looking at. So the sort of blue-green spongy things that you're seeing all over the place, those are the enzymes. So on the left side of this, I know you can't see my arrow, but on the left side of this, actually I'll just go back and now you can see my arrow. So this blue-green thing in the background, that's the enzyme. So that is an enzyme called fumarase. All enzymes end in ase. Good way to know him. And the fumarase enzyme takes this, I think this is isocitrate, and changes it into a very similar sugar. It just rearranges it a little bit is all it does. So this is the sugar. This is the substrate here, and this is the enzyme. And then there's a bunch of water molecules floating around. Okay? So in the presence of an enzyme, this molecule, this substrate actually has a place to go. It has an environment that pulls it apart properly, and it happens very fast. Over here on the right side is the uncatalyzed reaction. So there's no blue-green enzyme over here. There's no active site for this sugar. Here it is right here, and then there's a bunch of water molecules behind it. The only way it's going to react is if it's spontaneously, if there's enough energy or heat around it for things to bump into it just the right way, for it to change into whatever else it can change into. So that's very much due to random chance. There's no active site that provides a place for that to happen, or strains the bonds, or allows things to be much less random. So that's why the uncatalyzed reaction you're going to see is barely discernible. It's very, very slow. And that's not fast enough for a living thing. Our cells are very greedy. We need our energy quickly. We need our digestion reactions to happen fast. If we didn't have enzymes, this big blue-green enzyme here to help it happen fast, we really wouldn't be here.
reactions are millions to trillions times faster than those of the same reactions uncatalyzed. To speed up a reaction in the absence of enzyme, additional energy would need to be provided, heat, which jostles the substrates and occasionally provides enough energy to trigger a reaction. In the course of most reactions, an unstable and highly energetic transition state is formed as the substrates are transformed into products. An enzyme acts as a template for the reaction, binding to its substrate and holding it in the proper position to form the product. An enzyme also surrounds its substrate with reactive groups that stabilize the transition state, making it easier for the reaction to occur.
Okay, remember all those energy graphs we talked about? This is another energy graph. But this showed the difference between that big orange enzyme thing that looks like something out of Coraline, right? Came up, and what did it do? It provided an environment, right? It provided an environment for the reactants, the little smiley face guys that were on the left side, to fit into, to get pulled apart, and then become the happy little products, right? So that's why the blue curve that you see on the screen now is lower than the really tall curve that it was when there was no enzyme. And that is a reduction in activation energy. We learned about that last time. Okay, so let's see how much you remember from last time. Chat me in. Is this reaction exergonic or endergonic? Is it exergonic or endergonic? Based on the curve. What do you think? Chat me that. Yeah, Yes, very good. Yes, and Katie, you're also correct. It would also be exothermic, most likely. Yes, a little bit. You would release a little bit of heat here. But exergonic is correct. Energy is being released. We don't know what the reaction is, so we don't know how much heat would be released versus stored energy. But yes, because the reactants have more energy than the products, so even regardless of the activation energy humps, the delta G is negative here, and that means exergonic. Okay, I'm going to keep
to occur. To understand how enzymes work, let's take a closer look at a reaction in the citric acid cycle that is catalyzed by the enzyme aconitase. Aconitase binds to its substrate citrate and removes a hydroxyl group and a hydrogen atom to form the intermediate cis-aconitate. It then adds the hydrogen and hydroxyl back in slightly different positions to form the product isocitrate. In the active site, some amino acids are perfectly positioned to recognize the substrate and hold it in the optimal position for the reaction to begin. Some amino position to recognize the substrate and hold it in the optimal position.
Okay, so here's some college level chemistry for you. We're right at the molecular level here. So all this gray stuff around here, that is the enzyme's string of amino acids, right? It's three-dimensional protein shape, which is a string of amino acids. And then the black and blue parts sticking out are the R groups, different amino acids that are in the pocket of that active site and creating the chemistry of the active site that's going to allow this isocitrate molecule to get pulled apart and rearranged. So the blue-black parts are the R groups sticking out that are receiving that substrate, right? This yellow thing at the top is actually an iron sulfate complex. That's part of one of the R groups. And the red and black and white molecule is the actual substrate that's going to get pulled apart and rearranged. And let me show you how that works.
For the reaction to begin. Some amino acids are involved in recognizing and holding the substrate. Other amino acids are directly involved in catalysis. Histidine 101 acts as an acid by donating its proton. Thanks to the chemical environment of serine 642, it can act as a base by accepting the proton from the substrate. The active site of aconitase also contains an iron-sulfur cluster that stabilizes the substrate electrostatically and helps to position it relative to the catalytic residues. The first step in the reaction is dehydration. In this step, histidine acts as an acid and protonates the hydroxyl on the substrate, allowing it to leave as a water molecule. Serine then acts as a base, extracting a hydrogen atom from the opposite side of the substrate, forming the intermediate cis-aconitate. Cis-aconitate then flips upside down, and the complementary hydration reaction is performed. In this step, histidine grabs a hydrogen atom from a passing water molecule, placing the resulting hydroxyl group back on the substrate. Serine then returns its hydrogen atom, and the final product isocitrate is released. Notice that the enzyme itself was not changed by the reaction. It extracted a hydroxyl group and a hydrogen atom, and then put them back, starting and ending in the same state. This is the hallmark of a catalyst. When it finishes a reaction, it is ready for the next, so it can perform thousands of reactions in a row. Notice also that the shape of an active site is often flexible. Many enzymes surround their substrates, enclosing them to form the perfect environment for a reaction. Enzymes are fundamental to life on Earth, working every second of every day to maintain life processes in every cell in every living creature on the planet.